Hello everyone and welcome to my presentation. In this opportunity, I'm gonna talk about the potential risk of interspecific hybridization in ex-situ collections. My name is Sissi Lozada Gobilard and I'm a visiting postdoc at the Institute of Botany in Prague. Before starting, I would like to thank Hannah, Bojana and Susanna for giving me the chance to work with this amazing dataset. Let's begin. Ex-situ hybridization is the interbreeding of two species in artificial facilities such as botanical gardens. This spontaneous hybridization can cause negative effects. For instance, changes in life history traits such as germination. Like in this study, hybrids presented a higher germination rate compared to their wild parentals. Also, hybridization can cause loss of adaptation, in particular for a vulnerable endemic species highly adapted to extreme environmental conditions. This could contaminate ex-situ material that will be used for reintroduction purposes. If hybrids are fitter, they could outcompete the parental species and cause their local extinction in the wild. Changes in dispersal traits can also cause negative effects on populations. We know that changes in seed morphology can affect the dispersal ability. But how seed dispersal traits change between hybrids and parental species in ex-situ facilities has been overlooked. In this study, we focused on Minuartes makali. In the last decade, conservation efforts focus on this species, including creation of ex-situ collections for restoration purposes. Minuartes makali is an herbaceous species endemic to the Czech Republic that grows in highly specialized serpentine rocky soils, and their populations have been declining due to habitat fragmentation caused by anthropogenic activities since 1960. A related species, Minuartia caspitosa, has a broader distribution, occurring not only in Czech Republic but also in Germany and Belgium. This species grows in sandy soils, and it is classified as endangered in Czech Republic, and therefore has been grown in ex-situ facilities together with Minuartes makali. These two species are very similar morphologically, and the only way to distinguish them is based on seed. Minuartia caspitosa possess a much smoother seed coat compared to Minuartes makali. Even though these two species do not co-occur in nature, they can come together in ex-situ facilities and might be able to hybridize. This could have negative consequences for the restoration programs if hybrids are introduced by mistake in nature. Which leads us to our main question. We would like to know the effects of ex-situ hybridization in Minuartes makali compared to hybrids of Minuartes makali and Minuartia caspitosa in different traits related to fitness, seed morphology, and seed dispersal. In addition, we would like to know the effects of ex-situ hybridization on species adaptation. We hypothesize that hybrids will be fitter, will present a different seed morphology that would potentially affect their seed dispersal. But hybrids might also lose the ability to adapt to highly specialized serpentine habitats. To do so, we collected seeds from these two species and grew them in the garden. Then, each species was used as pollen donor for intra- and interspecific crossings. As a result of those crossings, we count the number of seeds. A set of these seeds were then used for germination assays and further grown to adult plants. Another set of these seeds was analyzed under the electron microscope to characterize its morphology, estimating the area and perimeter of the seeds. Approximately 6 months later, from the adult plants, we also measured seed dispersal traits, seed release height, and terminal velocity, which in combination with wind data and vegetation height from the natural populations were used in a deterministic model to estimate seed dispersal distances. And finally, we performed an experiment under different soil treatments, serpentine and sand, mimicking the natural conditions of Minoartis maecali and Minoartia caspitosa respectively, and with different light treatments, open and shade. We evaluated plant performance measuring the area they occupied and the number of flowers they produced. What did we find? The number of seeds was slightly higher in Minoartia maecali compared to hybrids, but not significant. But germination rate was significantly higher in hybrids. Similar results were previously reported in other studies. Increased fitness in hybrids is known as heterosis. This process mainly occurs in the first generation, as it was observed in our study. These results suggest that if hybrids are introduced by mistake to the wild, they might be able to spread rapidly. However, we do not know if higher fitness can also be maintained in subsequent generations. This is something to focus on in the future. With respect to seed dispersal, we calculated seed release height and terminal velocity and used these values in a model to estimate seed dispersal distances. We found a significantly higher maximal dispersal distance, almost double in hybrids compared to Minoartis maecali. Respect to seed morphology, seeds of hybrids and Minoartis maecali did not differ in either area nor perimeter, making it impossible to distinguish them only based on taxonomy. And finally, our experiment showed that under open light treatments, there was a significantly higher number of flowers produced in hybrids compared to Minoartis maecali in both kinds of soil. This was in contrast to our expectations. However, in the shade treatment, hybrids produce fewer flowers, especially in sandy soils. Because of this, hybrids did not do better in all treatments. There might not be a heterosis effect. But we can say that hybrids perform better in extreme open serpentine conditions. One thing that we did not include in the experiment is draw, which is very important in extreme serpentine habitats. It will be very interesting to test how hybrids will do under such conditions. To summarize, we did not find differences in seed production or morphology between Minoartis maecali and hybrids. But germination, seed dispersal, and plant performance, at least in open serpentine conditions, were higher in the hybrids. We can conclude that hybrids are potential risk because they might have higher ability to colonize and overtake Minoartis maecali wild populations if introduced by mistake, in particular in serpentine habitats. To achieve a successful restoration of Minoartis maecali in the wild, hybridization events should be avoided, and therefore we recommend to keep ex-situ collections of Minoartis maecali and Minoartia caspitosa physically separated as much as possible. With this, I conclude my presentation. Thank you very much for your attention, and I am up for questions.